Reproduction in bacteria. Bacteria reproduce both asexually and sexually. We first talk about asexual reproduction. Asexual reproduction in bacteria occurs by the following methods. First is binary fission. This is the most common type of asexual reproduction in actively growing bacteria and occurs during favorable conditions. On this basis, bacteria were once called fission fungi or schizomycetes. In this process, the cytoplasm and the nucleoid divide equally into two without mitosis, and the two daughter cells formed are identical to each other, hence the name binary fission. The whole process of binary fission involves two steps: genome replication and septum formation. Both the events occur simultaneously and are triggered by a mesosome if present. Binary fission begins with DNA replication. Now, how does this DNA replication take place? Well, DNA replication starts from an origin of replication which opens up into a bubble. The replication bubble separates the two DNA strands. Each strand acts as a template for synthesis of a daughter strand. The DNA replication is bidirectional, starting from the point of origin and resulting in the formation of two circular daughter DNA molecules. The zooglia stage. What actually is the zooglia? Well, under unfavorable conditions, the bacterial cells come to lie in chains and get surrounded by a lot of mucilage. This stage where the bacteria come to form chains is the zooglia stage. As you can see in the graphics, the bacterial chains, or now you may call them the zooglia. Conidia formation. The terminal portions of bacterial filaments cut off rounded structures in chains are called conidia. Conidia on detachment form new individuals. Here we have those terminal portions of bacterial filaments in chains or conidia, as may be illustrated in the case of Mycelioptera. The gonidia formation. First, the gonidia. The bacterial cells produce a number of flagellated daughter cells within. These daughter cells are also called as swarmers. The cell wall of parent cell ruptures, releasing what come to be called as gonidia. This type of reproduction is reported in Rhizobium species and also in Volvox carteri. Cyst formation. In this type of asexual reproduction, the parent cell secretes a highly protective covering called the cyst wall, which can resist unfavorable conditions. On return of the favorable conditions, the cyst wall dissolves and the bacterium gets released. This cyst formation helps in perennation. This is reported in Entamoeba histolytica. And here is the cyst formation that we are talking about. Budding. Some bacteria continuously produce protrusions called buds, which on detachment form new individuals. Hypomicrobium vulgare and Rhodomicrobium vanillae are common examples of budding bacteria. Now you can have a clear picture in the case of Planktomyces. Look at these terminal ends as the buds are sprouting. It is on the detachment of those buds that the bacterial forms can reproduce. fragmentation. It occurs in colonial cyanobacteria. After reaching a certain length, bacterium breaks up into pieces called fragments. Each fragment is the beginning of a new colony, as may be seen in the case of yeasts. In this case, a Saccharomyces species, after reaching a certain length, breaks up into fragments. Echinates. These are formed in many cyanobacteria. Echinates are thick-walled resistant cells with abundant food reserves, which are frequently developed singly next to the heterocyst. However, their position is variable, as in the case of Anabaemia. Endospore formation. When the environmental conditions are adverse, some gram-positive bacteria, especially the bacilli and certain blue-green bacteria, produce thick-walled resistant spores called the endospores. During the endospore formation, a portion of the cytoplasm and a copy of the bacterial chromosome dehydrate and get encased by a very thick wall. The rest of the cytoplasm and the bacterial cell wall deteriorate so that the endospore is released. The thick wall of a typical endospore is distinguishable into 4 parts, namely the exosporium, the spore coat, the cortex, and the core wall. Sexual reproduction or genetic recombination in bacteria. Typical sexual reproduction through the agency of gametes is absent in bacteria. There is no fertilization and meiosis. However, the gene transfer in bacteria occurs by 3 methods: conjugation, transformation, and transduction. First, conjugation. What is conjugation? Well, a mechanism resembling sexual reproduction was discovered by Joshua Lederberg and Edward Tatum in 1946 in the bacterium E. coli. It was confirmed by Hayes and Vollmann in the same bacterium. They found that physical contact was involved in conjugation between the 2 conjugates, and the DNA from one is transferred into another through a conjugation tube. It involves a transfer of DNA from a donor or a male cell to a recipient or a female cell through a specialized sex peleus or the conjugation tube. This is how the transfer of DNA takes place. It takes place from a male cell to a recipient or a female cell, and this is the conjugation tube we are talking about. Mechanism of conjugation. The cells that have the capacity to serve as donors during conjugation are differentiated by the presence of specialized cell surface appendages called F. pili. In the bacterium, conjugation can occur in several ways. Some important examples are conjugation between the F plus male and the F minus female. The E. coli shows 2 strains, one acting as a donor, that is the F plus male, and the other as the recipient, the F minus female. Conjugation between high frequency recombination male and F minus female. The F factor can integrate into the host chromosome. An F plus cell carrying an integrated F factor is called an HFR, which stands for high frequency recombination. Therefore, the F plus male becomes the HFR male. In the integrated state, the F factor mediates the transfer of a chromosome of the HFR male cell to a recipient, which is the F minus cell. Now we talk about transduction. N. Zinder and J. Lederberg in 1952 observed in Salmonella typhimurium, a mouse typhoid bacterium, the transfer of DNA from a donor cell to a recipient cell by bacteriophages. This is what came to be called as transduction. In most cases, only a small segment of the host DNA is transferred. In the graphics, you can see the DNA transfer from a donor to a recipient cell by bacteriophages. Here is the bacteriophage. Now we talk about types of transduction. Two types of transduction can be distinguished: generalized transduction, which can transfer any part of the host DNA, and specialized transduction, which is restricted to the transfer of specific DNA segments. In certain cases, bacterial DNA is injected by a phage, but it does not replicate. This kind of transduction is referred to as abortive transduction. In generalized transduction, a random or nearly random segment of bacterial DNA is wrapped up during phage maturation in place of or along with the phage chromosome in a few progeny particles called transducing particles. Generalized transducing phase can therefore transport any gene of the donor cell to the recipient cell. In specialized transduction, a recombination event involving the host chromosome and the phage chromosome occurs, producing a phage chromosome containing a segment of bacterial DNA. Specialized transducing particles thus always contain both phage and bacterial DNA. Specialized transduction is so named because a given virus transduces only genetic markers of the host that are located on one small region of the bacterial chromosome. Specialized transduction is mediated by the temperate bacteriophages, whose chromosomes are able to integrate at one or a few specified attachment sites on the host chromosome. In its integrated stage, the phage chromosome is called the prophage. The chromosome of temperate phages of this type are thus capable of both autonomous replication, that is, replication independent of the replication of the host chromosome, and integrated replication, which is the replication as a segment of the host chromosome. As such, they are examples of genetic elements called episomes. Transformation. Frederick Griffith, an English bacteriologist, accidentally found that the heat-killed bacteria of the virulent strain of Pneumococcus pneumoniae could transfer characteristics of its strain to the non-virulent strain of living bacteria. Avery, MacLeod and McCarty observed that it is due to the transfer of DNA segments from the dead cells to the living cells they called the uptake and incorporation of DNA by bacteria as transformation, and the normal ability to take up exogenous DNA from the environment as competence. Therefore, transformation may be defined as the gene transfer by soluble or naked DNA which has been extracted or otherwise liberated from a donor bacterium to a recipient bacterium. This is the longest known and historically the most important kind of gene transfer in bacteria.